little early, and if anyone has some questions, we can go over that. But let's make sure we get through our material first. So to remind you of the difference of meiosis, if you haven't thought about this for a while, the purpose of mitosis, first of all, was to take actually I'll write it like this, a diploid cell, and keep in mind, we're talking about the difference between somatic cells and germline cells. That's not a term we use a lot, but a somatic cell is sometimes called a body cell to mean any cell that is not sperm or egg. Okay, so those cells are not somatic cells, those are germline cells, but all the rest of our cells in our body are somatic cells. So we take a diploid somatic cell, which has 2 copies of all our chromosomes, right? And a good way to think about diploid cells is to call them 2N. And N is going to be a number we're going to throw around a lot when we talk about meiosis and genetics in general. But N is a variable that people use for the haploid number of chromosomes. All right, so if you are a human, which I think you are, your haploid number of chromosomes is 23, so your somatic diploid cells would have 2 times that, right? So each of your somatic cells would have 46 chromosomes, all right? And the reason that we use N there instead of just saying 46 is that if we use N, this can reference any organism. Like if we were talking about dogs, they would not have 46 chromosomes. Dogs have, what's 2 times 39? 78 chromosomes. So if we just say 2N, this applies to any organism that is diploid as opposed to putting a specific number. All right, so if we go through mitosis then, we end up with, let's say it this way. We start with 1 somatic cell and we end up with 2N diploid cells. All right, so we basically end with the same thing we started with, just twice as much. Meiosis, on the other hand, first of all, has 2 separate division stages. So we have meiosis 1 and meiosis 2. In meiosis, we start with a 2N diploid germline cell. We go through meiosis 1, and after going through the first meiosis, we end up with 2N, I'll put it in parentheses there so it doesn't look like a part of a word, 2N haploid cells. So after the first division, we are already haploid, and that's strange because, well, I'll get to why it's strange in a minute. So after meiosis 1, then we go through a second meiosis, which is meiosis 2. And after meiosis 2, we end up with 4 haploid cells. And these haploid cells are referred to as gametes. So those are the sperm or the egg cells that would be used to in sexual reproduction to produce another organism. Okay, so 2 divisions, and we go from diploid to haploid. So we call meiosis a reduction division. Even though we're making more cells, we're reducing the number of chromosomes. Okay, and if we look at an overview of this, we start out with a diploid cell, and this cell just has a single set of chromosomes. So if this was a human cell, we'd have 23 times what we're looking at there. But that single set of chromosomes undergoes replication prior to meiosis, so that all the chromosomes have their sister chromatids, right? And all their sister chromatids then go through the first round of replication to produce 2 daughter cells, but the sister chromatids stay together, right? In meiosis, the sister chromatids separate and we're done. But here the sister chromatids stay together, and then they go through a second meiosis, the sister chromatids split, and we end up with 4 haploid cells, and those are going to be either, as we said, the sperm or the egg. Okay, so a little bit different. So let's take a look at a couple of the places where meiosis differs. In large part, actually, do I have a full, yeah. In large part, it's very similar to mitosis. So let's actually go ahead and say that first. We have a prophase, a prometaphase, a metaphase, a anaphase, a telophase, just like mitosis, bless you. And then we have it again, prophase 2, metaphase 2, or prometaphase 2, metaphase 2, anaphase 2, telophase 2. So the names of the phases are the same, and if you look at what's happening, most of the things in those phases are also the same. The nucleus delaminates and disassembles, the chromosomes condense, they line up at the metaphase plate, they get separated, we have the cleavage furrow. So if you're not counting the chromosomes, that looks just like mitosis. Then it happens again. The nucleus has reformed, but now in the second meiosis, it's going to delaminate again. The chromosomes line up at the metaphase plate, they separate, and we have cytokinesis a second time. So in large part, it looks the same. The main difference is that the sister chromatids don't split up in the first meiosis. They do split up in the second. And the second major difference is that in the first meiosis, the chromosomes find their match. And that's where we'll talk about one of the big differences. So in meiosis, not mitosis, only in meiosis, there is an event that we want to discuss called crossing over. And this happens in prophase 1, one of the first things that happens. Okay, and when crossing over happens, the chromosome 1 that you got from your father and the chromosome 1 that you got from your mother are going to find each other, okay? And those are called homologs. And so, or sometimes they're called homologous chromosomes, but a homologous chromosome and a homolog are the same thing, just different names for the same thing. So, let's see. When this happens, the homologs, which again are the matching chromosomes from each parent, line up and attach by forming, this is a nice word, a great spelling bee word. They line up and attach by forming a synaptonemal complex. Synaptonemal complex. Okay, so synapse means for two things to come together, so that's where that word root comes from. So we don't see this in mitosis, okay? This is strictly a meiotic event. So they line up and form a synaptonemal complex, and that synaptonemal complex looks like this. They're proteins pulling the two chromosomes together, and they're matching up with their partner, and they're matching up by sequence, which means that the same DNA sequence on your dad's chromosome is matched up with the same DNA sequence on your mom's chromosome. The reason that matters, the reason that's important, is they are going to swap pieces. All right, so the swapping of the pieces is what's called crossing over. So they line up and attach by forming a synaptonemal complex, and then the homologs exchange equivalent segments with each other. It's a really strange thing on the surface. Why would they be doing this? So they exchange equivalent segments, and in the diagram up there, it shows one little area where they swap, but actually it will cross up, two homologs will cross over multiple places along their length. So this is underrepresentation of what's really happening. But when they exchange equivalent pieces, the first question would be, why? And the answer to that is it generates diversity. This is one of the most important parts about sexual reproduction, which is that if we didn't undergo sexual reproduction and meiotic cell division, the chromosomes you inherit from your parents would be the exact chromosomes you would pass on to your offspring, which would be fine if those chromosomes were fine. But what if there's a problem? We would like to have some diversity generated in every generation so that we're continually evolving and adapting to our environment. So by exchanging pieces, you're creating a chromosome that never existed before. So this mostly red but a little bit of blue chromosome is not your parent's chromosome, right? It's part of their chromosomes mixed together. So there might be combinations of genes that didn't exist before, right? So in nature, organisms that do not cross over, like bacteria, that simply go through binary fission and split apart into two new bacterial cells, without going through crossing over, they have no chance to evolve or adapt to their environment unless they just undergo mutations. So it's a whole different scheme. It's really one of the most positive attributes of being a sexually reproducing organism. Okay? And then after the crossing over happens, the cells actually, the chromosomes, not the cells, stay attached for a little while at a place they call the chiasma. So the chiasma is the site of attachment. So move over here. And so you can see this microscopically. If you're looking at a cell, you can actually see where the chromosomes have exchanged parts. So this isn't like speculative or just what we think happens. You can see this happening. So a chiasma, and the plural of that, which is what you see more often, is chiasmata, is the site of crossing over. It's the place where it happens. All right, so that's one of the major diversified events. If you take genetics, which many of you might, we'll talk about that in much more detail, but that will be okay for us in this class. And another thing to mention is that if we were to scan around nature and look at different things like animals, fungi, some plants, we would see that they are doing some different things than us. We're not going to go on a deep dive into each of those different organisms. You would if you take zoology, which is a great class, highly recommended. But just to point out, the way we do it is not the universal way. So we have an egg which is haploid. It fertilizes, the sperm fertilizes the egg to make a diploid zygote. The zygote's just the fertilized egg, which divides and eventually makes our little organism. But other organisms do it differently. So fungus, for example, have a part of their life where they are haploid even though they're somatic cells. So those aren't their gametes. They also have reproductive cells, which like their somatic cells are haploid. They briefly have a diploid zygote, which can then form spores, which again are haploid. So they have sort of an inversion of the way we do it. They're mostly haploid, sometimes diploid. We're mostly diploid, very briefly haploid. And then ferns are doing all kinds of weird things. They've got a diploid zygote and a diploid plant, but they have haploid spores, and then they briefly have a haploid plant, which then makes sperm, and then we're diploid again. So they're all wacky there. But I'm holding you responsible for what the animals are doing. That's the one we're focusing on. I'll leave these topics for our zoology teachers to bring to light. Okay, so in a given organism, just a couple more things about chromosomes, and then we'll switch over to our other topic today. If an organism is diploid, as we said, that means they have 2 copies of every chromosome. So this is a fruit fly, just because it's easy to see with its 4 chromosome sets. But there's the the variable we used, 2n, right? Meaning 2 sets, and so that's diploid. Now, it's possible to be more than diploid. So if we have 3 sets, it could be triploid. If you have 4 sets, it could be tetraploid. Now, these conditions don't occur in humans. They can occur in some animals. A lot of amphibians, for example, are tetraploid. A lot of frogs are tetraploid. And they do just fine. Like, it's not an abnormality, it's just that's how they evolved. Some animals can be triploid, even though they're not normally triploid, and still be fine. For example, if you happen to have a farm pond and you stock it with grass carp to control the vegetation, those grass carp are triploid. Not because they're triploid in nature, but because the hatchery that created those grass carp by law has to make them triploid so that they're sterile. So a triploid grass carp cannot reproduce in a pond, which is great because if they did, they would be an ecological disaster because they would outgrow all the other fish in the pond, they would eat all the vegetation, they would spill out into a creek and then get into a river, then wreck that ecosystem. So grass carp are not animals that we want to have in our waterways. They're not native and they do a lot of damage. So they're useful, but only if we can keep them from breeding which is why we make them triploid. They're still fine, their biology is mostly normal, but they can't breed. Okay, I'm going to skip the color vision thing since we're on a little bit of a time crunch today. One thing that can happen during meiosis is we can end up with something called aneuploidy. And aneuploidy is a general term that refers to an abnormal chromosome number. Okay, so instead of having a complete extra set, which would be triploidy or tetraploidy or any type of polyploidy, aneuploidy is referring to just the number of chromosomes, and usually one extra or missing one or a couple extra. And this happens when the chromosomes that are supposed to be splitting apart during anaphase, right? The microtubules reach out, they grab the kinetochores, and they pull either the homologs apart or later they pull the sister chromatids apart. But if that doesn't work, it's called non-disjunction, right? That's exactly what the word sounds like. They don't disjoint, they don't come apart. It's kind of a double negative. And so aneuploidy results from non-disjunction of chromosomes. So in either of the anaphases, when we're trying to pull either the homologs or the sister chromatids apart, if they don't come apart, too many are going to go to one side and too few are going to go to the other. All right, now you've heard, undoubtedly, you've heard some of these aneuploid syndromes. I'm going to ask you, just for the sake of focusing your studying, I'm going to ask you about the sex chromosomal ones. Instead of asking you about all of them, I really just focused on these on the exam. That way you can reduce the amount of memory you have to commit to this table. But I'm sure you've heard of Down syndrome. Down syndrome is the most common aneuploidy. And if we designate autosomal versus sex chromosomal, autosomal means the non-sex chromosomes. That's a term you hear when we talk about inheritance a lot. But it simply means the non-sex chromosomes. So for humans, 1 through 22. So we can have, our voice breaking, going through some changes. We can have autosomal aneuploidies, and most of the time, this doesn't develop to a live birth. So if you were to analyze, well, we talked about this with karyotypes, I think, when we were talking about how to do a karyotype. Didn't we talk about how to do a karyotype?
Yeah.
Why were we talking about that? Oh, at the beginning of mitosis. When you analyze miscarriages, usually it's because of an abnormal chromosome number. So most of the time, we don't see aneuploidies make it to a live birth. But when they do, there are only 3 autosomal aneuploidies that occur with any type of frequency. The others are so rare that we won't even mention them on the table. But the 3 that do occur, the most common one is a 21st trisomy, which is called Down syndrome commonly. That was named after the doctor that first described it, Sir John Langdon Hayden Down, back in the 1860s, actually. He described it long before he knew what caused it, but in the 1960s, scientists learned that it was a 21st chromosome trisomy. So 3 copies of chromosome 21, and this results in a number of different characteristics, which, as I said, you're probably familiar with. That's associated with Down syndrome. Some of them are premature aging, intellectual disabilities. They tend to have short stature. They have an increased risk for Alzheimer's and leukemia, which is not a good scenario. But those guys do much better now than they used to, and a big change in the way that society treats individuals with intellectual disabilities has been that in the last 50 or so years, people stopped institutionalizing them. So if we were having this class in 1965, for example, or even 1970, you probably wouldn't have known or ever seen or been around many people with Down syndrome, because oftentimes they were sent to live in an institution away from the rest of society. And what we learned from that terrible experiment is that people do not fare well under those conditions. And those individuals had shortened lifespans, shortened quality of life, all kinds of things which are negative. So fortunately, the trend in society has been to be much more accepting of people who are different from themselves. Okay, trisomy 18 and 13, a much more tragic scenario. If you have an extra 18th or 13th chromosome, the chance of dying before your first birthday is about 90%. So these guys really, really struggle with the first year of their life, mainly because of heart defects, but they have a lot of other physical impairments that make that first year really, really difficult. So two very tragic situations, Edward and Patau syndrome. Sex chromosomal aneuploidies are quite distinct from autosomal aneuploidies for a couple reasons. One is that the body tends to tolerate an extra sex chromosome much better than it tolerates an extra autosome. So for example, an extra X chromosome in an individual that has a Y chromosome results in a syndrome called Klinefelter syndrome. And this is a super brief synopsis of what those individuals have, but Klinefelter syndrome results in individuals who have underdeveloped gonad tissues, so they do not sexually mature, and so the main phenotype or characteristic is that they're infertile. And the rest of the characteristics they may encounter are often so mild that people don't know they have Klinefelter syndrome until they try to have offspring and can't. And then they are tested and find out, lo and behold, they have an extra X chromosome they never knew about, but had been living life, otherwise normally. Breast swelling is not apparent in all individuals with Klinefelter syndrome. It does occur in some, which puts them at risk for breast cancer, which is an unusual scenario for males because it's almost absent in the rest of the male population. I think of what else. They tend to have sparse body and facial hair, but again, that is so different from person to person. It's not like diagnostic. No one says, oh, you don't grow a thick beard, you must have an extra X chromosome, right? That's not true. And so again, the condition is often the characteristic so mild that people don't know it until they find that they are unable to produce viable sperm. XYY, this is another condition. Look at the characteristic, tall. All right, so again, tend not to notice that you have an extra Y chromosome unless you're tested for another reason. Sometimes individuals with XYY have some learning disabilities associated with that, but that again doesn't distinguish them from the rest of society, right? That happens in all of the human population. A triple X, same thing, tall, right? Again, menstrual irregularity, also common in the population at large, and so those individuals both of these individuals are fertile. So again, unless they have another reason to test or do a karyotype, they may go through their entire life and never realize they have one more chromosome than many of the people around them. The last one, Turner syndrome, is, this one does have some fairly significant characteristics. So short stature, they tend to grow much smaller or slower than their peers. They do not develop a functional ovarian tissue. So at a time when their age-matched cohort would be going through puberty, they don't. And if their doctor hasn't recognized it before then, that's when they would, when they don't go through menstruation and they don't have the secondary sexual characteristics, which would be breast development, hip development, redistribution of body fat. And so those individuals, again, usually diagnosed earlier, but if they don't enter puberty, then that's when they would know. And with Turner syndrome, a solution to some of the characteristics, if you consider those characteristics needing a solution, is hormone replacement therapy. But that doesn't go back in time and cause their ovaries development. So they are not going to be sexually fertile even if you give them hormone replacement. But the hormone replacement therapy can reverse some of the other characteristics that might be psychologically important to those individuals. So pretty complicated scenario. Okay, so that's the end of that section. Let's put that to rest and bring up our final topic, which looks longer than it is. Some of the slides we've actually already looked at. But the final topic is going to be cancer, which is what's the right word? A loss of cell cycle control is one way to think of cancer, which is why we're talking about it at this time. And cancer results always from mutations to our DNA. Okay, a lot of other things play into the development of cancer, but the fundamental thing, the thing that starts it all, is mutation. And mutation can be caused by lots of different things. We'll look at a few of those in a second. But where the mutation occurs is important. So if we look at our naked male, I'm not sure why it was important for this diagram that he's completely nude. He could have just as well worn clothes, but anyway, there we are. We're in biology, so might as well make it provocative enough to keep your interest. But if you have a mutation, let's look at our little embryo over here who's getting this laser zap to his brain. If we have a mutation that is not in the tissue that is going to form our gametes. So let's say you overexpose yourself to UV light in a tanning bed, or you tan too often and you get a mutation in some of the cells on your skin, and that mutation develops into a tumor. That's not going to be passed on to your offspring, right? It's not a good situation, especially not for you, and hopefully it shouldn't make this so personal. It's not a good situation for a person, but hopefully, you're treated and you're okay. But the main point is your gametes do not carry that mutation, right? Because it didn't happen in cells that produce egg or sperm. On the other hand, if you have a mutation in the cells that become your gamete producing cells, that will then get passed on. And so there's a really important distinction between mutations that occur to us during our lifetime, that are ours alone, and mutations that occur in our gamete cells. So we could pass a mutation from generation to generation to generation if it's in the gametes. But mutations that we acquire in our lifetime are ours to bear. Okay, this is a little bit forward thinking, since we haven't talked about DNA and amino acids yet in the context of how proteins are made. But hopefully we have enough context to understand this, at least for now. And then we'll talk about it in more detail in our last unit. But the point of this slide was to discuss the types of mutations that can occur that would affect potentially a cell's ability to function normally. So one type of mutation, if you remember from maybe sometime in your past when you talked about DNA in an old biology course, you may remember that, right, first one up, GCC, GG. You may remember that DNA encodes our proteins by using 3 nucleotide segments. Okay, and so each of these 3 nucleotide segments is called a codon, and the codon is going to indicate which amino acid goes into the protein. So when a protein is made, it will have amino acids based on the instructions in our DNA. So this protein, whatever it's supposed to be, hypothetically, has a methionine because it had an ATG codon telling the ribosome to add a methionine. It then adds an alanine because it has GCC. It then adds a glycine because it has GGC, then a proline, CCG, and so on. So each of these 3 groups of nucleotides tells the ribosome, this is what you should add to this growing protein. All right, we'll talk about that in great detail in the next couple of weeks, but for the moment, I just want you to understand these instructions convey this information. And if that information changes, the protein is going to change. Well, potentially can change. And so there are 4 types of mutations that we want to know just for now, for this exam. The first one is called a silent mutation. Silent mutations can alter a nucleotide without altering what happens in the protein. Okay? And the reason for that is maybe something we'll discuss later. I'm going to put what's the right word? You say you're going to do something later. Put a blank in that. A what? Put a pin in it. Yes, thank you. So we'll put a pin in that. But for now, let's just look at what these do. So the first one is silent, and so that is a change where the protein outcome is not affected. Not that you have a choice when you're mutated, but if you did, this is the one you would choose, right? This doesn't change anything in the outcome. So all of us have differences between us in how our proteins are encoded because of silent mutations, but we're still making the same proteins. So these types of mutations just happen in the natural world and don't affect any outcome. The second one is called a missense mutation. And this leads to a different amino acid. So the codon changes from encoding one amino acid to encoding another. So that can be drastic. If it's only one amino acid, you might think, well, what's the big deal? The protein has 1,000 amino acids. What will happen if we just change one? Sometimes something drastic will happen. It just depends on which amino acid changes. They're there for a reason. So that can be a big deal. The third one is called a nonsense mutation. And our codons, including the role of indicating which amino acid should be added to the growing protein, they also have to tell the ribosome to stop. They have to say, okay, this is where the instructions end. You're done with the protein, move on to the next thing. And so a stop codon can exist at the end of a protein, but a mutation can happen that changes a codon that used to be something else, like lysine, to a stop codon. So that means that it's telling the ribosome to stop too early. So this leads to a stop codon in the wrong place. Okay, and then the last one is called a frame shift. And a frame shift is a really drastic mutation. If we think about the DNA encoding amino acids in groups of 3s, for example, if you delete or add a nucleotide to the DNA, which can happen, think about what the consequence of that would be. So if we had this encoding these specific amino acids corresponding to GCC, GGC, GAC, etc., what if we lost a nucleotide? Right? If that happened because of a mutation, now our code would look like this. This would have to shift over there. This would have to shift over. This would have to shift over, and so forth. So these two codes no longer encode the same protein, not at all. Everything past the frame shift is off, all right? And so that's a really drastic change. So a frame shift leads to a different protein sequence after the mutation. So everything from the mutation on is what I mean by after the mutation. Everything from that point forward gets all jacked up, to use a common phrase. Okay, so all that stuff we will dive into in great detail in coming weeks, but for today we just want to know mutations happen, they can have these results, and we'll have an outcome in the protein that is not usually favorable. One example of a single mutation in a single amino acid change that can have a drastic effect is sickle cell anemia. In sickle cell anemia, one change to one amino acid causes your cells, which are normally nice and round and buoyant and holding lots of oxygen, your red blood cells, to form the shape of a sickle, which is like a C shape. And this does not bind oxygen very well. So the sickle cell anemia is caused the anemia is caused when you have cells not binding oxygen very well and blocking capillaries. So instead of moving smoothly through the capillaries and distributing the oxygen they're holding, they're less capable of the oxygen potential, and they end up piling up and blocking blood flow. It's so a very dangerous disorder. And the point of the example is it was just a single nucleotide difference that caused a single amino acid, and that was enough. That was enough to cause a tragic disorder. Okay, so why is our DNA mutating? What would cause it? Lots of different things. There are chemical causes. Well, let's look at the easy ones. Physical causes, X-rays. So when you have X-rays, that is mutating your DNA. That's why you don't have X-rays every day. That's why if you work in a clinic that gives X-rays, you wear lead protective clothing because X-rays can't penetrate lead. And you also wear a little badge that detects the amount of X-ray exposure you've had, and that badge is checked every now and then to make sure you haven't gone past the presumed allowable amount. And so sometimes X-rays, of course, are used intentionally to mutate and kill cells. If you had a large tumor, people used to use X-rays. It's not as common now as it used to be, to try and shrink the tumor by actually causing massive DNA mutation. But the downside of that, of course, is you're mutating other cells too. So it has a lot of collateral damage. UV light, which we cannot possibly avoid and shouldn't avoid because it helps our skin produce vitamin D, which we need, but excessive UV light causes something called pyrimidine dimers, and this mutation can lead to cellular damage, which can lead to tumors, which can then become cancer. So our UV light exposure should be pretty minimal, really, and certainly you should not go out of your way to have extra UV light, like a tanning bed. That is just asking for problems down the road. Okay, what about chemicals? Some of these you're probably never going to experience. I don't know that you'll be dealing with any nitrous acid or nitrogen mustard, hopefully not. But those are chemicals that are known to cause DNA damage. The reason they're listed here is because people sometimes use them intentionally to cause damage. So a way that people used to study disorders in mice, I mean, they weren't studying it because they cared about the mice. They were studying mice because it reflected on what happened in other organisms like humans. But the way they would study it is they would intentionally expose mice to things like ethyl methane sulfonate, and they were trying to mutate the mice. And then they might end up with a mouse that had a disorder that mimicked diabetes, or a disorder that mimicked some other degenerative disorder, maybe like Alzheimer's. And so they were creating what people call mouse models to replicate disorders that occur in humans. And to do that, they were just mutating the mice randomly, hoping they would create this syndrome, which is kind of sad for the mouse. Well, not kind of sad, very sad. One that you might indeed be exposed to is benzoapyrene. Does anyone know why you would be exposed to that? Where does benzoapyrene occur? Where would you be exposed to that? Smoking. Yes, good call. So cigarette smoke, which hopefully you do not and will not ever do, and marijuana smoke, which that's up to you. I'm not going to give a public statement on that. Those are two things that produce benzoapyrene. So if you are consuming either of those, what happens when those organic molecules combust, because you're burning them and then sucking the smoke down, you are sucking down benzoapyrene, which is a byproduct of that combustion. And benzoapyrene causes mutations in your DNA, which is why smoking is a known cause of lung cancer. And marijuana use has probably not been documented long enough in long-term studies to see how its effect on lung cancer will be. But it releases the same chemical at higher quantities, because most people who smoke marijuana are not smoking them in a filtered cigarette, right? They're just smoking it straight up. And so you're not filtering out a lot of that benzoapyrene, you're just sucking it straight down. And furthermore, you're holding it in your mouth or your lungs usually. You're not like blowing it out too quick. So bad. I guess I did make a statement after all. Okay, we don't need to worry about that guy. So what can we do to fix this? Well, one thing we can do is rely on our cells to repair DNA. So the type of DNA repair I'm interested in you knowing for the case of this particular exam is, do I have this on the, how it is repaired? Yeah. The one that I think you should focus on is this one, nucleotide excision repair. Oh boy, it's harder than it looks. Which is where the abnormality is recognized, a portion of the strand is removed, and the strand is then replaced with normal DNA. And so we do repair and fix our DNA when mutations happen. The problem is we don't fix them all. We're not 100% perfect in our ability to repair DNA. And so if you are out exposing yourself to a lot of UV light, for example, in tanning beds, for a period, you will be able to fix those mutations you're causing in your skin, but at some point, your skin cannot keep up with the amount of mutations. Your DNA repair mechanisms are only so good, right? You can think of it like patching a hole in a boat. If there's a hole and you can patch it, then you're fine. If there's another, you can patch it, then you're fine. But another and another and another, eventually you're gonna sink, right? So you can only do so much. And nucleotide repair is one way to do that. This is just an example of how it works. You don't need to memorize these steps, but just to show you what takes place when you have a mutation like that, to fix it, you have to actually cut it out. And so you have enzymes that find it, enzymes that cut the DNA and remove it, right? They literally remove it, and then they just replace it with a new section. So there's no fixing most mutations where they are. You have to just get rid of them and start fresh. So your body can do this, but only for so long. And there's a disorder, which I ask about on the review, called xeroderma pigmentosum. And xeroderma pigmentosum is what this poor little guy has. It's quite a mouthful here, xeroderma pigmentosum. But this disorder is caused by the inability to repair DNA. And so in xeroderma pigmentosum, this is a lack of DNA repair enzymes, leading to the expected outcome, which is multiple tumors, especially on the skin because of the UV light, but also internally. So they cannot fix DNA. And by not being able to fix DNA, they develop multiple tumors, multiple malignancies, and this, of course, can lead to a shortened lifespan because of all the small, multiple cancers that they're dealing with. So a really horrible situation for those guys. All right, let's talk about cancer now directly. So we've been kind of skirting around the issue here so far, but let's now dive in. And one thing that is important to know about cancer is that it is a stepwise progression. I mean, you don't just wake up one day and the cancer is there. It happens over a period of time in a sequence of events. And so we can call, let's see, what would be a good way? Let's just call it progression of cancer. So cancer is going to begin when there is a mutation in a cell, actually multiple mutations in a cell that lead to that cell behaving abnormally. And specifically, it's growing too fast. So let me see if I put terms on that. Progression from benign, okay. And so the first step we can call hyperplasia, which means excessive cell division. Excessive cell division. And this doesn't necessarily mean that cancer is going to happen, but it does mean that something's wrong in a cell and it's dividing more than it should. The second step is the development of a tumor itself, which we can call a benign growth, and that has a specific meaning. I'm going to go ahead and call it benign tumor. And the reason we call it a benign tumor is that the cells have not invaded neighboring tissues. Cells have not invaded neighboring tissues. So you could have a huge benign tumor, but if it doesn't invade the tissues that are around it, it's still just a benign tumor. So that name doesn't indicate the size or the location or what that tumor might be doing. It just means specifically it hasn't invaded neighboring tissues, right? And then the third one, or the third stage here, is called a malignant tumor or malignant growth, either one. And this is not a good development in the progression of the tumor, because this malignant growth indicates that instead of just growing and staying in its own place, even though it's growing, it invades tissues around it. So invades neighboring tissues. That means the cell has taken on characteristics, the cells have taken on characteristics they didn't have before. And if it's able to invade nearby tissues, this indicates that it's probably going to be a problem, almost certainly going to be a problem. And so the last stage then, which is when words you never want to hear, is when it becomes metastatic. Or I'll use the noun version, metastasis. So in metastasis, tumor cells are going to break off the original tumor, infiltrate the circulatory system, and go to other parts of the body. I'll say cells from tumor enter the circulatory system. This is one reason when a tumor is located in an individual, one of the first things that they do is they look at the nearby lymph nodes to see if they have tumor cells, because that would indicate that the tumor has left its original location, and the lymph nodes and your blood vessels are all intertwined, right? All the circulatory system it interacts and it is intertwined. So if they've gotten into your lymph nodes, that means they are moving around the body, but the lymph nodes collect cells. And so you can't just, drain a person's entire blood system and look for the tumor, but you can look at the lymph nodes, because the tumor will have likely lodged some of its cells there. Okay, let's keep going. This is just a close-up of it. I don't think we see anything different there. Okay, so why would the tumor be dividing excessively in the first place? Well, we saw this already. So we talked about the MAP kinase pathway at the beginning of our mitosis section. So this is not new information, I'm just re-showing the slide. But one of the things that can happen when cancer progresses is that this system, which is receiving a signal and transmitting the signal to the nucleus, and the nucleus then produces proteins that lead to cell division. Oops. That's normal, right? There's nothing wrong with this. The problem in cancer is that this starts to go more than it should. Those tumor cells start to activate this pathway when they're not supposed to. So, in other words, the proteins are behaving in ways they shouldn't. So how does this relate back to some of our terms? Well, cancer is the product of DNA mutation, as we said. And that mutation can affect the cell differently depending on which genes get mutated. And so there are two different types of genes associated with cancer. So I'm going to call them cancer-related genes. And the first group are genes called, let's see, just say it a different way. The first group are called oncogenes. Okay, and in our chart it says examples of genes of growth factor pathways that can become oncogenes. Okay, so that implies that oncogenes are not the normal scenario, which is true, And so an oncogene, the way we can define it is that these are mutated versions of proto-oncogenes. Okay, which means we'll need to define what proto-oncogenes is. But an oncogene is the mutated version. This is the bad version. This can lead to, as the chart says, unregulated cell division. So mutated versions of proto-oncogenes, actually, let me finish my thought here. Mutated versions of proto-oncogenes that lead to excessive cell division. Okay, so this gives a clue about what a proto-oncogene might be. A proto-oncogene, I'll write it up here, is a normal gene involved in regulating cell division. So a normal gene that's involved in regulating cell division might be something like the growth factor, the gene that encodes the growth factor, the gene that encodes the ligand, the gene that encodes some of the kinases, the gene that encodes RAS. So all of these things are normal proteins. We have to have our cells divide, but if those proteins are mutated, we then call them oncogenes instead of proto-oncogenes, which are normal. We call them oncogenes, and these lead to excessive cell division. So anything that might normally be acting as a growth factor or a growth factor receptor or something relating that signal to the nucleus, if you have too much of it, that's a problem, okay? And one of the proteins that we already looked at last time was RAS. So in this diagram, RAS, remember, is early on. So this would have been like a day and a half's notes ago. But we talked about how RAS can be active or inactive depending on whether it's attached to a GDP or a GTP. And RAS is one protein that sometimes gets mutated so that instead of being controlled and turned on and off, it's just always on. Okay, it's always on. In fact, the name RAS came from the place where it was discovered, which was a rat sarcoma, which is a tumor that was found in a rat, and they were studying that rat to understand why, and so they named the protein that they found RAS. Okay, now there can be other things that lead to tumor formation or cancer. Let me make sure I'm following my instructions here. Oh, I want to put another thing on my list before we talk about that guy. So oncogenes, that's one cancer-related genes category. The second category are genes called tumor suppressor genes. So to keep this straight here, oncogenes are things we don't want because they're mutated versions of our normal genes. The normal genes that they correspond to were called proto-oncogenes, which are things we do want because they regulate the cell cycle. When they're mutated, they do bad things. Okay. The second one, tumor suppressor genes, these are genes we definitely want. Tumor suppressor genes regulate the cell cycle, repair DNA, and initiate apoptosis in damaged cells. Well, to be accurate, let me say this. Tumor suppressor genes encode proteins that do these things. The genes don't do these things. The genes encode the proteins do the work. So these are all good things, right? We want to regulate our cell cycle. We want to repair DNA. We want to initiate apoptosis in damaged cells. So these are good things. All right, so let's look at some examples of how when these genes fail, we have problems. So in the case of some leukemias, for example, we have genes that normally regulate cell division, like ABL and BCR. And if chromosomes actually exchange pieces, not in crossing over, which is a good thing, but just a dysregulated cell breakage where they then try to put the pieces back together wrong, sometimes you get chromosomes that shouldn't exist, right? Those genes should not be next to each other. They're on different chromosomes. But scientists have found that a lot of different cancers, especially blood cancers, occur when genes from different chromosomes get matched up next to each other to create a new sort of, well, they call it a fusion or a chimeric gene. Chimeric is a reference to a chimera, which is, I believe it's Greek mythology, where you have two different animals in one, like the human torso on a horse, for example. But the point of it is chromosomes breaking, which can happen because of radiation, for example, can lead to the cell trying to fix things and it fixes them wrong, and you end up creating oncogenes from genes that normally regulated cell division, but now in this new mutated form do not. And so that's a cause of a specific type of blood cancer. We also don't have tumor suppressor genes. Oh, there we go. Let's look at this slide first. I'm a little out of order. Some tumor suppressor genes that are really important. Some examples, I'll give you three main examples to know. One is called P53. Not a very exciting name, but the name corresponds to its molecular weight. They named it before they knew what it did. But this is like the mother of all tumor suppressor genes. It promotes DNA repair, prevents progression through the cell cycle, and promotes apoptosis. It does the three things that define tumor suppressor genes, right? It's so important. In fact, most cancers that actually become malignant and metastasize, if you check them, which sometimes people do to understand how to treat them, they will have a mutated P53. So for a cancer to be successful, in other words, you have to take this gene out, all right, because it does these things which would otherwise prevent the cancer from developing. A second gene, which I'll use as an example, is RB. And RB was the first tumor suppressor recognized. I don't think I have a picture, no. But RB stands for retinoblastoma. And so this gene and the resulting protein that it makes regulate the cell cycle so if you have a mutated version of this, your cell cycle will not be regulated. And this was discovered, as the name might give you a clue, in eye cancers, and specifically in eye cancers of children. Most children do not have cancers. It's uncommon as a childhood disease, but retinoblastoma is one of the exceptions of a cancer that occurs during some children's early years. And so this was discovered in eye cancers. So the children had a mutated retinoblastoma protein, which led to retinoblastomas, and oftentimes that cannot be fixed. You have to just, you lose the eye, which is really sad for those kids. And then the third one is BRCA1 and 2, and I'll put it over here. BRCA1 and BRCA2. And these acronyms stand for breast cancer associated. It doesn't say on our table there. Breast cancer associated. And so the role of BRCA1 and BRCA2 normally is to repair DNA damage. And the repair of DNA damage is essential for all cellular health, but these genes specifically seem to predispose they do predispose an individual to breast cancer when they're mutated. And so when these guys are mutated, I'll say it this way, mutation in these genes raises the risk of breast cancer from about, what's 1/8? 12.5%? So it raises the risk of breast cancer from 12%, which is the lifetime risk for a female human. So the females in their lifetime have a 1 in 8 chance, or 1 in 8 females in their lifetime will have breast cancer. So that's a 12% risk. But if you have a mutation in BRCA1 or 2, this raises the risk from about 12% to, or I'll put it this way, greater than 50%. And so this discovery, really changed the way people thought about cancer, because cancer is not something you can inherit, but you can inherit the risk, right? You can inherit a predisposition. And so if you inherit a mutation in BRCA1 and 2, you have a greater than 50% risk of cancer versus the general population. And so those cases where families know they have that mutation, oftentimes people will take different preventative steps, have more frequent exams. Some people who know they have a greater than 50% chance of breast cancer take the step of actually having a preventive mastectomy, meaning later in life they have their breast surgically removed before a diagnosis is even made. And so that's a really big ethical question to ask and to think about, certainly beyond the realm of this lecture. But in any case, those are 3 examples, P53, RB, and CDK2. And we talked about the CDKs and the cyclins a while back when we were talking about cell cycles and how the cyclins rise in abundance and then fall, and they regulate the cyclin-dependent kinases. P53 and RB are involved in this process specifically as the cell is going through the cycle before it synthesizes the DNA. Our friend P53, our main cellular protector, can halt cell division while it tries to repair DNA. And if it's not able to be fixed, P53 will do its other role, which will be to kill the cell. So without P53, we wouldn't be able to recognize the DNA damage, fix the DNA damage, and kill the cells where the DNA is damaged. So it's so important. Okay, and then I think that we want to look at viruses. That's on my list here, the role of viruses in cancer. The main virus that is not on this list, strangely, that is worth thinking about associated with cancer, viruses as a whole are not living things, right? A virus is not a living organism. It can't reproduce by itself. It's not like a cell. It doesn't have a nucleus. What a virus does is it infects a cell, and then it gets the cell to make more of itself, right? So when a virus gets inside of a cell, it tells the cell, hey, I've got DNA, replicate it, make my proteins, and then usually as a thank you, the virus kills the cell on the way out. But that being said, the virus wants cells to divide, because when cells divide, they make DNA, and if the virus can jump in on that bandwagon, more virus will be made. And so viruses often are associated with cancer. And the virus that I would want you to know about is the human papillomavirus, or HPV. Now this virus, as a sexually transmitted disease, can lead to, in a non-cancerous situation, genital warts, which are in themselves not good. But because it's causing excessive cell division, sometimes this HPV virus can cause cervical cancer. So this is noteworthy for that case alone. But it's also noteworthy because it's the first example of a cancer for which there's a vaccine, right? This was a totally novel concept when I was first teaching genetics, because the vaccine was developed in the early 2000s. So I was already teaching and talking about cancer and the fear of human papillomavirus, which was transmitted at high rates, especially on college campuses, and led to cervical cancer in many cases. And then a vaccine was made, and that was a crazy idea that you could vaccinate yourself against the risk of cancer. But that vaccine now is routinely given to adolescents. I don't know all the names, the brand names now. Gardasil was one of the names of that vaccine that you may or may not have had. But lots of other cancers are known to cause lots of other viruses are known to cause cancers in other organisms as well. In fact, the chicken cancer was the first one discovered. At that point, people thought, well, maybe now we know what causes all cancers, because this was like in the early 1900s. But of course, that turned out not to be true. Okay, then the last part of this we've already done. I went ahead and skipped to cell death earlier. I was just feeling it for some reason. So we've actually already completed that. So that is the end of all the material that you are responsible for